Hello, I'm Stefano del Prato, and I am very much indebted to Professor Canat for this invitation, which once again I can accept only as a virtual. As a virtual. Let me start again. Huh? Hello, I'm Stefano del Prato. I'm Emeritus Professor at the University of Pisa, and I'm very much grateful to Professor Canat for this new renewed invitation to participate in this meeting, although once again I'll be participated only virtually. What Professor Kanata has asked me to speak about you is shown here in the slides of the title, which is the evolution of insulin therapy from human insulin to smart insulins. These are my conflict of interest. And let me just, briefly bring you through what has been an amazing history of the evolution of the insulin formulation for the treatment of diabetes. Here in saying type 2 diabetes, but this is also works for any form of diabetes. You may remember insulin was discovered back in 1922 by Banting and Best and all the team in Toronto in Canada with the development of animal insulin. But after that time, after that incredible, amazing time of the discovery of insulin, insulin has provided a new formulation and a new surprise and new opportunities very often throughout the history. And you can see here a sort of a condensed narrative of what has been the evolution of insulin. We move from animal insulin to human insulin to rapid insulin to a new formulation of basal insulin to end up with the most recent formulation of insulin, which is the once weekly insulin. These insulins are the so-called icodec insulin and F-Citora, and they have a specific structure and specific characteristics. The F-Citora insulin is a fragment crystallizable fusion protein which links a single chain protein which is fused through a peptide linker to a modified human IgG2. It has a reduced insulin receptor affinity with full agonism for the receptor and a selectivity against the human IGF-1 receptor and functional properties similar to native insulin, and with an half-life of 17 days, which allow the use as well as a once-week insulin injection. Insulin icodec, on the contrary, is an acylation with icosahexaenoic acid, which is an insulin analog acetylated with a C20 fatty diacid side chain with a reduced, similar to what has been for F-Citora, affinity for insulin receptor, and a reduced insulin receptor mediated clearance and enzymatic degradation, which allows a mean half-life of 8 days, once again compatible for once weekly administration. These are the two human insulin that are currently either available, icodec, or are very much in advanced development phase, F-Citora. And what I'm going to do here is just to really summarize what is the result of the clinical trials that have explored the safety and efficacy of these two formulations of insulin. You can see here a summary of the program Onward, which is the clinical development program for the insulin icodec, and which has been tested in people with type 2 diabetes, where insulin naive to treatment. In type 2 diabetic individuals treated with insulin, so a switch trial. In insulin-naive participants with a type 2 diabetes, again versus degludec as compared to the previous one, which was versus glargine, and in type 2 diabetic individuals treated with glargine, and once again in those that have been treated with both formulation of insulin. And you can see here overall what has been shown in this clinical data, in these clinical results, is that this once weekly insulin injection, as compared to the daily basal insulin injection, is associated with a overall similar reduction, if not better reduction in A1C, with a trivial increase or trivial occurrence of hypoglycemic events, and with a slightly improvement, which is not very much different from what can be observed with the traditional insulin, daily insulin injection in the time in range for glucose control. This insulin formulation has been also evaluated in people with type 1 diabetes, and you can see here the results. Now, once again, here you can see that this once weekly administration of insulin is very much as effective as the daily insulin injection, with the only difference that the rate of hypoglycemia, particularly during the first phase of the titration, is greater with the insulin icodec as compared to the insulin with a once daily insulin injection, in this specific case, degludec. As I said, the insulin icodec is currently available in the market in many countries all around the world, while in an advanced phase of development, clinical development, is the insulin Fcitora. And what has been presented here, available, is these two trials that have been performed in people with type 2 diabetes, either naive to insulin or already on insulin treatment, with the type 2 diabetes as compared to insulin degludec once a day. And what you can see here once again is that overall the improvement in glycemic control that can be achieved with the insulin once weekly is similar to the one that can be achieved with the intensification of the insulin injection once daily. This is not associated with significant increase, as you can see here, in the hypoglycemic episodes, and is associated with a slight and not significant different improvement in the time in range among these subjects. Along with these two trials that has been performed in people with type 2 diabetes, also trials being performed in people with type 1 diabetes, very much alike what has been reported also with insulin icodec. There was, once again, a similar improvement in glycemic control with an increase in the hypoglycemic events, particularly during the time of the titration of the insulin. So there has been a progression toward new formulation of insulin, maybe to make the life easier for people with the diabetes, moving from 365 injection a year of basal insulin to 52 injection a year. But with along with the improvement and with the quest for long-acting insulin, there's been also the continuous quest for more rapid activating insulin that can be used at the time of a meal. There are different ways to improve the absorption of regular insulin into the bloodstream. There are different formulations that have been used. You can see here just an example. But this example here has been evaluated in different ways, and what we have been made available with, or some of the attempts that's been performed, are represented here. So we have the faster insulin aspart, the ultra-rapid insulin, and the biochaperone insulin that actually has not been achieved the market yet. But what I like to point out here, what is the attempt that has been tried to achieve, is really to increase the rapidity of the absorption of insulin. And you can see here in this very early phase after the injection of this insulin, the solid line to raise at a quicker rate as compared to the regular insulin that have been made available so far. Now, this more rapid appearance into the bloodstream of insulin is associated with a more rapid effect in terms of the pharmacodynamics. You can see here the glucose infusion required to maintain normal glycemia after the injection. And once again, you can see a more rapid need to increase the insulin infusion, suggesting that this rapid increase in the circulating levels of insulin is also associated with a more rapid action of the insulin. And this translates into a potential better postprandial glycemic control, as indicated here, respectively, for the faster aspart insulin, as well as the ultra-rapid Lispro and the biochaperone Lispro. So this is a modification on the structure of the insulin that has been used to improve the rapidity of the action and absorption, but there are multiple different ways to really improve this rate of absorption. For instance, there have been attempted to try to improve the local absorption by using chemical or physical factors, as indicated here, to increase the absorption. But also there have been also the opportunity to use a different way to deliver that insulin, taking advantage of a more rapid passage from into the bloodstream. And one example is the pulmonary. As you can imagine, the vascularization of the lung is quite complex and quite complex and large. The surface of absorption is very, very large. It has been compared to the size of a tennis court. And because of that, the lung has been quite an interesting target since the beginning of the discovery of insulin. Insulin was discovered in 1922, but already in 1925, we had the initial attempt to really provide insulin through inhalation. And you can see here the experiment. And in the attempt to really to do this into the clinical ground, there has been a continuous evolution, although this evolution has been made not that simple by the need of a bulk instrument, as you can see here, but also you may remember the exuberant instrument, the technosphere, and so on and so forth. And actually, the technosphere is the one that is currently available, not in the European market, but definitely in the American market. The technosphere is a particular formulation of powder insulin that allow a very rapid absorption of insulin through the lung. As you can see here, comparing the pharmacokinetics and the pharmacodynamics of the technosphere insulin as compared to the Lispro insulin as an example of rapid insulin that we currently use in our clinical activities. The question is, to which extent this change in the pharmacokinetics of this rapid insulin can translate into an advantage. This has been evaluated in a number of trials that have been summarized here in this meta-analysis, that already this meta-analysis has been performed 10 years ago, but recently also confirmed, at least as far as type 1 diabetic individuals is concerned, in a more recent meta-analysis. But by and large, what it has been shown is that in people with type 2 diabetes, as well as in people with type 1 diabetes, there is a slight advantage or at least no disadvantage at all as compared to the regular insulin. So because of this, as I said, the Technosphere insulin is available in the United States, although the FDA has established this warning for the potential risk of acute bronchospasm in patients with chronic lung disease, and also alerting for the need to keep a close eye on the potential growth activity exerted by insulin in the lung and the liver, although the most recent results have not indicated any particular risk in the risk of the pulmonary malignancy. Now, the lung also brings up another issue concerning what is the evolution of insulin, which is, alternative routes of insulin delivery. I already mentioned the lung, but you can see here that many others have been evaluated: the nasal, the buccal, the oral, the transdermal route. But out of these different approaches, the one that attracted more interest, obviously, is the oral enteric route. As you can imagine, ingesting insulin is much easier than injecting insulin. Although the oral route represents a major challenge because of the degradation of a hormone like insulin in the stomach due to the low pH, the local pH, and because of the action of the proteolytic enzymes, and also the limited permeability across the gastrointestinal epithelium. So it has been considered that insulin, as it is, when it is administered, if it would be administered by oral ingestion, will have a very, very low bioavailability, which has been accounted for more or less than 0.01%. So alternative approaches have been used to overcome these difficulties in administering insulin through the oral way. One possibility is just to ingest insulin, but have that insulin to bypass completely the stomach and really open up and delivering insulin into the early segment of the intestine, in particular in the duodenum, as it can be achieved by the use of intestinal micropatches, as represented here. These are capsules that are pH sensitive. They are not degraded into the gastric environment, but once they reach the intestinal lumen, they open up to deliver this patch that have the ability to adhere to the intestinal epithelium and then to deliver insulin. And this is what has been adopted as an approach by this formulation of insulin that has been tested initially in some clinical experiment. You can see this oral insulin, which is the ORMD 0801, showing definitely a potential efficacy in terms of nighttime glucose, 24-hour glucose, fasting plasma glucose concentration, overall daytime glucose. So this insulin has been proven to exert the expected glucose lowering effect. Although more clinical trial is not really allowed to achieve a significant and meaningful effect. And what has been presented, at least in this press release by the company producing or developing this oral insulin, was that the primary endpoint of mean change from baseline in A1C after 26 of a week of treatment was not met, as well as, there was also some problem with the secondary endpoints. And it was really a sort of a disappointment that has been released by the chief executive officer when he said that the outcome were very disappointing given the positive result of prior trials as the one that I showed you a moment ago. This doesn't mean that there is no hope for an oral insulin, because the same company has recently acquired quite a lot of a big investment to try to move into phase 3. So more will have to wait to learn about these possibilities. There are other options, however, to really facilitate the assumption that the ingestion of the oral insulin and to make that insulin to work. As I said, one way is to bypass the gastric area completely, or the other one is to really improve the local absorption of insulin to allow the insulin to really bypass, to go through the gastric mucosa. An example of that is what has been achieved by developing the oral GLP-1 receptor agonist, like semaglutide, that has been linked to this sodium aminocaprylate, which allows the local preservation of the molecule, and in the meantime also facilitate the transfer across the mucosa of the molecule. And this same approach has been used, is currently used for GLP-1 receptor agonists, as I mentioned, but also being attempted also with insulin. And here is an example of comparing the efficacy of oral basal insulin versus insulin glargine. And you can see a very nice response, a very much similar result in terms of reduction in the fasting plasma glucose concentration throughout this 8 week of treatment with this oral insulin as compared to glargine, as well as in terms of the reduction of the daily glucose concentration. The problem is that the program apparently has been stopped because the amount of insulin that was required to provide sufficient activity and bioavailability, it was so large that would have been requiring a major effort in terms of production of this insulin. But what is interesting here is the concept of going through the gastric mucosa. And this can be done by chemical solution like this one, or it can be done mechanically. And this is an interesting proposal. These small pills here, which have the shape of a turtle, are something that has been developed in order to allow these pills containing insulin. And these pills have a specific shape. It's a turtle shape, as I mentioned, because they roll in a very quick time, and they also end up with the base of this capsule adhering with the mucosa. And once that adhesion has occurred, there is a small spring within this capsule that are able to inject through the mucosa. So once again, achieving the systemic circulation and delivering insulin. And here's a proof of concept. What you can see here is that the plasma insulin concentration and the area under curve of the plasma insulin concentration and the reduction in plasma glucose concentration in terms of percent change and in terms of changes 2 hours after an oral glucose load in these animals that have been given this device. Now, this is just, of course, as proof of concept. As far as I know, this approach is still under development, but I thought that it was of interest for you to know different way to really proceed. And there is different way to proceed and different way to really provide injection of insulin, as it has been demonstrated throughout the history of the development of insulin. The major breakthrough really was represented by the insulin pen, but now these pens are becoming smart pen. And this smart pen can be linked directly to glucose monitoring and can generate a flow of information that can be translated into a feedback information to remind the time for the injection of insulin and also to improve the overall clinical management of insulin administration. Just as an example, this is a recent paper that has been published looking at the effect of this system, either active, so people were able to receive a feedback from the system, or mask, not receiving the feedback, in people with type 1 diabetes with a poor glycemic control. And you can see here the result in terms of the time in range, the significant improvement in those receiving active feedback, the significant reduction in the estimated A1C, the reduction in the time above range, and the reduction in the very high glucose concentration. So overall suggesting that an assisted process and assisted the management of the insulin treatment may result, particularly in people with poor glycemic control, into an improvement in that glycemic control. But of course, the expectation is really to provide and to confer intelligence and to the system that is able to automatically deliver insulin. We now live in the time of the automated insulin delivery system that are based on the continuous glucose monitoring and the use of an algorithm that control the insulin delivery. But even this system that seems intelligent enough is going to improve over the time, both in terms of the delivery with the pump moving from outside to inside, most likely in the peritoneal area, with an improvement also in terms of the glycemic, the glucose monitoring, that it may become more and more calibration free, and even using artificial intelligence or the machine learning to really adapt the algorithm to the individual need of the individuals. And with that also, the expectation is really to come to a condition so we can think of forgetting diabetes. I had the opportunity to collaborate with some of the scientists here in Pisa with a big expertise in nano systems, and they have and we have tried to elaborate on the concept to have a continuous glucose monitoring, which is recording the glucose concentration, of course, and also communicating with an insulin pump, which is positioned very close and adherent to the jejunum. And then on the basis of the glucose reading, of course, as expected, that pump will deliver insulin. Allow the control of the glucose concentration, even in a more physiological manner, because the insulin will be delivered into the peritoneal system, and because of that, also restoring the portosystemic gradient. This, of course, will require insulin to be used. And also, what we have been elaborating and tested in animals is the refilling of the pump through a magnetic peel that can travel through the stomach and really dock in front of the pump, and the pump can then absorb insulin, highly concentrated insulin, so refurbishing the insulin reservoir of the pump. So in other words, there will be no need to really open up the body to replenish the insulin. And because of that, the system can go on and go on. And of course, the capsule can be delivered through the natural ways. And the other point is that the other point that we've been facing was how can we then maintain the power of the pump. But that was the easiest way, because as you can recharge your mobile using magnetic systems, the same can be done with a magnetic belt here that can be able to recharge the pump. So it's a big future, but a big future that really include insulin, but also insulin that becomes responsive through mechanical system, but also an insulin that becomes smart by itself, becoming responsive to the glucose change, as it can be achieved by using either polymer matrix-based glucose responsive insulin, or also by increasing the system that can increase the permeability during hyperglycemia. Here is just an example of that. What has been done here was to generate a polymer which is sensitive to the local pH, and the local pH can be changed as an effect of the activity of the glucose oxidase, and the glucose oxidase, changing the milieu, result in the bioreduction of these polymers that can open up, and these polymers that contain insulin release the insulin. And of course, with the glucose going down, there will be less reaction to the local glucooxidase that allow the reconstitution of the polymer and then the trapping of the insulin, so modulating the insulin activity. And this can be achieved and can be administered using multiple micro needles, as is shown here, with a transdermal transcutaneous administration. Does it work? Preclinical data suggests that by and large it seems to work. You can see here in streptozotis in diabetic mice the effect of this insulin delivery system with a glucose-sensitive response of the insulin in terms of reduction in glucose as compared to different conditions, and in terms of the tolerance to an IV glucose tolerance test, and also the change in glucose concentration after administration of this array patch. So there are multiple other ways to provide this kind of insulin delivery. But also the other option is that rather than using polymer and working with a system that encompass the insulin and open up the insulin and make that available to the biological system, also to elaborate and to work on specific molecular glucose responsive insulin. So insulin molecule which conformation that made them be able to interact with the hormone, with the receptor or insulin receptor as a function of the glucose concentration. And here's just an example. In this case, the insulin is conjugate with a carbohydrate, and the carbohydrate can bind an lectin, an lectin receptor mediated that leads to degradation of this insulin complex. So when the glucose is high, there will be more affinity of the insulin to the insulin receptor. When the glucose is low, there will be more affinity of this insulin to this lectin receptor that leads to degradation. So made the insulin available as a function of the prevalent glucose concentration. Now, I just try to summarize for you what is the future here for the insulin, but just to give you an idea about the smart insulin, because as I can recognize from the title of my topic, that was the leading target for the future formulation of insulin. This is a sort of a time map of what has been the development or the search or the research in terms of the glucose responsive insulin. You can see here that all started almost 40 years ago, and there's been a long pathway with more to go before we'll be able to really have an available smart insulin in our hands to treat people with type 2 diabetes. And you can see the different approaches have been used over the time with the glucose, the glucose oxidase based and the polymer based one, being representing the most likely to achieve the clinical setting. So what I try to do in this very short time is really summarize what has been a phenomenal journey of insulin. It has been a journey that started off in 1922, but it looks to me that it's a journey that is not at the end yet. And I think that after 100 years, we can still look at insulin as a young product, although more and more indication and more specific indication for the use of insulin needed to be adopted in people with type 2 diabetes on one side, but also in people with type 1 diabetes as well. And I thank you very much for your attention.